Okay. Grazie ragazzi, willkommen and bienvenue to Cannabiscienzia's LinkedIn Live for this week. To those of you joining us live, thank you for being here with us today. And for those of you who will be checking out the recording at a later time, also a big thank you to you. We hope you enjoy it and perhaps you leave this session having learned something new. Cannabiscientia aims to combat the bottlenecks of the medical cannabis industry using the power of education and through the training of healthcare professionals on the effective use of cannabinoid medicines. And in doing so, we believe we are effectively increasing access to medical cannabis for patients worldwide. On the screen right now, as you can tell, there is an overview of let me go back. First, here are our services, what we do. So you can scan our QR code and get in touch with us. And I was too excited. So that's why I skip ahead into this because we recently just launched our handbook, Principle of Clinical Cannabinology, that just happened on the 2nd of May in beautiful London. And amongst other things, it covers the endocannabinoid system, clinical practices, regulatory landscapes, and available products in all the different jurisdictions in Europe. So, of course, if you're interested in learning more about the handbook, reserve your copy, or learn more about sponsorship opportunities, please scan the QR code now on your screen and get in touch with us. But enough with all of this, because today we will be discussing different pharmacological ways to modulate the endocannabinoid system with one of our amazing science committee members. Thank you for being here, Dr. Ines Reynoso, who is a scientist working at the University of Bern. So Ines, thanks again for being with us. Yes, and I'll just leave the floor to you so you can give a better introduction about yourself.
Thank you. I mean, as you mentioned, I am a postdoc researcher at the University of Bern. I'm working at the Gersh Group Lab. We do quite a multidisciplinary research, mostly based on the endocannabinoid system. We do in vivo work, working in animal models, mostly mice, but also analytical chemistry, how the endocannabinoid levels are changed in the different models we evaluate, also in vivo work, so meaning cells, immune cells, neuronal cells, and also other a few things that we do here. I did my PhD in Mexico, and this was mostly associated to neuroscience, but from the beginning I was working with the endocannabinoid system, so I have about 10 years experience working in the field, and I'm happy to be here. Thank you.
Thank you for being here. And today I also have my amazing Dr. Fabio Turco, who will be following along with some of the most geeky, sciencey questions, because we really want to use this half an hour that we have today with Ines that is doing really interesting work. So without further ado, I will just pass the floor to Fabio. Thank you.
Okay, thank you, Viola. Hello, everybody, and welcome to this Cannabiscienzias live on LinkedIn. So, as Viola was saying, today we'll be exploring novel pharmacological approaches to modulate the endocannabinoid system with Ines Reynoso Moreno. That is, she's a postdoc from the University of Bern. So, Ines, as we know, cannabinoids, especially THC and CBD, are well-known compounds that can modulate the endocannabinoid system. But, however, there are other substances that can interact with the endocannabinoid system too. So, Ines, can you shed light on other compounds or substances apart from the classic cannabinoid that have the potential to modulate the endocannabinoid system?
Yeah, of course. I mean, for example, molecules like THC are what it's called agonists to the cannabinoid receptors. We have two main cannabinoid receptors, CB1, which is mostly in the CNS, and CB2, which is mostly in immune cells, but there is also like overlapping with the receptors are expressed. And agonists like THC, they bind to the receptor and activate a cascade of response that develop all the effects that we see through the modulation of these molecules. So these agonists are the classical cannabinoids because they directly act on the cannabinoid receptors. But as some of you might know, we also have the endocannabinoids, which are the agonists that our body produces all the time. And these are all the time activating the cannabinoid receptors to modulate many, many different processes, biological processes. These are, modulators of how our body works. And these are expressed in the whole body. So another key element to modulate the endocannabinoid system, it's also modulating the endocannabinoid levels because these are produced by our body in the moment that our body needs them. So they are on demand. So if I have pain in certain way, endocannabinoids will be released to modulate this pain response. So to modulate these endocannabinoids, there is two main ways to do it. One will be inhibiting their degradation. So meaning once the cannabinoids are produced, they are rapidly degraded, so they are not working anymore, so they have a limited action. And if we inhibit the enzymes that degrade these endocannabinoids, then we will have the endocannabinoids for longer time, and therefore we prolong how long these effects will be lasting. The other strategy is mostly based in the transport of the endocannabinoids because the degradation of the endocannabinoids occurs at the intracellular level, so inside the cells, and the endocannabinoids are released in the outside of the cells. So to get to the degradation enzymes, they have to pass the membrane and go inside the cells. And this is actually debated process because there is many ways in which it can happen. But if we can inhibit this passing of the endocannabinoids to the inside, so this transport, this is another way that we can prolong the effects of the endocannabinoids. I mean, at the end of the day, also because we are inhibiting the degradation because they don't get inside. They stay outside for longer, and after a certain period of time, they will also get inside and they will be degraded. So overall, this will be the two mainly other ways, additional ways that we have to modulate the endocannabinoid system. In this case, I mean, we have enzymes that are particularly associated to the degradation of one of the endocannabinoids, of each of the endocannabinoids. As some of you might know, we have two main endocannabinoids, which are anandamide, and the other one is 2-AG. And there is selective inhibitors for the enzymes that degrade each of them. So these are two different pharmacological approaches. And also endocannabinoids are present, each endocannabinoids can be present in a certain tissue in a more pronounced manner. So certain tissues have more anandamide, certain tissues have more 2-AG. For example, in the brain, there is way more 2-AG than anandamide. For example, here for CNS-associated disorders, it's probably better to modulate 2-AG levels. But also we have anandamide in the CNS. So for example, in those cases, modulate the levels of both endocannabinoids can be more beneficial than modulated only one. For example, here is where the inhibition of the uptake process, of the transport process, can be also much more beneficial of only inhibiting the degradation of one of the two endocannabinoids. Other way that maybe is also important to mention is the modulation of the ligands by positive allosteric modulation. The allosteric modulation will be not directly the ligand who binds to the receptor, but it's a molecule that binds in a different place and can prolong the effects of the ligands. And this can also be beneficial because can modulate more subtlety the way that the receptor is being activated. This is also still being a lot evaluated recently because it can be also quite promising. But that's overall, I think this will be the ways to modulate the endocannabinoid system. I hope that was clear.
Sorry, I switch and I come in because I'm really interested in what you just said, and I was wondering whether this way of, for example, inhibiting the transporter. I actually have a couple of questions that just came to mind,
I mean,
and I
this
wanted to.
transporter process is quite
Yeah,
a hard topic.
yes, it is.
Especially
But
in our
I
lab.
assume that as everything is complicated in the endocannabinoid system, that we have specific transporter for each endocannabinoid. So it's not going to be a generic transporter, or can we
I mean,
use a sort of?
actually, from what we have seen, will be the same protein that works for both endocannabinoids. It will be just one protein. I mean, actually, one of the main areas of research in our group is actually finding understanding this transport of endocannabinoids. We have been actually working quite a lot on this, and actually soon, hopefully, there will be the publication when one can understand, because maybe I'm getting a little bit ahead of myself, but I mean, we also have developed some inhibitors of this transport process. And before, some years ago, we actually didn't know which was the target, which was this, I will call it protein, that was responsible for inhibiting the transport of endocannabinoid. We have spent quite a lot of years understanding this process. We are every day getting a little bit more closer to how this works. I also don't want to confuse everyone with how complicated this process is, because it is actually quite complicated. But I mean, just saying this, from what we have seen, yeah, this protein can also modulate the transport of both endocannabinoids. That's why when we have done in vivo models, I mean, it's animal work, we can see that both endocannabinoids, anandamide and 2-AG, they both increase, which you cannot see, for example, when you are using just inhibitors of one of each enzyme.
Thank you. Actually, this totally, well, first of all, we're so looking forward to your upcoming publication. So this is a really interesting topic that I need updates on. So looking forward to that. But I was wondering whether, is, in your opinion, then having something that generically inhibits a transporter, so that generically inhibits two different, sometimes even almost opposite endocannabinoids like 2-AG and anandamide, is it safer somehow than inhibiting, for example, only the enzyme that breaks down? I'm talking with the overview of what happened years ago with the trials for the Phi inhibitor back
Yeah,
in Portugal. So I
yeah,
was
but
wondering,
yeah, sorry.
is there also a method of is this like a good way to improve safety of the application?
In a way, yes, it is actually. I mean, just to clarify, in case of this compound, the BIA compound, the Phi inhibitor that failed in the clinical trials, I mean, it was also one of the main questions to answer if the effects that they were seeing, I mean, this participant that unfortunately died, if this was really associated to Pha-associated effects, the Pha inhibition. And actually, after an extensive research, it was conclusively said that it's not associated to the Pha inhibition. So this was discarded. It's actually just because of off-target effects that were not seen before. That's why actually with all these novel molecules that have been developed and recently, it's very important to understand other off-target effects that can be relevant. And in terms of these, that the inhibition of the transport will increase both endocannabinoids, is actually not as something that it will be bad, because in terms of the inhibition of the enzymes, either for anandamide or for 2-AG, I mean, they actually increase the levels of either anandamide or 2-AG in very, very high levels. I mean, you can see up to 100 times higher levels than you will see in normal cases. And this can also be not really beneficial. This have seen particularly in a more clear way for 2-AG, because this can also lead to tolerance. I mean, there is so much 2-AG that the receptor is desensitized and there is no more response to the drug. This has not seen so much for anandamide, but for 2-AG, yes. And in case of the inhibition of the transport itself, I mean, the increase that we can see in both endocannabinoid levels is for too much up to twofold, but most likely always around 50%, 40%. So it's a more physiological increase. And it's really in a safer, much more safer way, if one wants to call it like this. Of course, further experiments are necessary to do. Also, soon one of them will be going to clinical trials. For so far, I mean, they are really not toxic.
Thank you. Okay, I leave it back to Fabio. Thank you for replying extensively to my question, actually.
No problem.
Okay, thank you, Viola. I was wondering about so as you discussed, we have various ways to modulate the endocannabinoid system apart from the classical cannabinoid. So we can use allosteric modulators, enzyme inhibitors, or these novel inhibitors of the transporter of the endocannabinoids. But according to you, which one of these compounds have the best potential to be to develop to be put in therapy regarding some kind of pathology?
I mean, at the end, all the different strategies might offer certain advantages, and also it could be related to which disease they might be used for. There have been already some clinical trials for anandamide inhibitors or inhibitors of the degradation of anandamide or 2-AG. So far, just some 5 inhibitors have been successful in the clinical trials, but nothing has yet come out of the market. In terms of course, for the transport inhibitors, it's too early to say. Soon we are starting to come with the clinical trials. I might also be biased because from what we have seen so far, our research tells us that it really is promising the use of inhibitors of the transport of endocannabinoids, because they increase both endocannabinoids in a more a slight manner, not so exacerbated increase, more physiological. There is also other advantages associated to other potential targets on the endocannabinoids they do have, because they not only activate the cannabinoid receptors, they also activate other receptors. So this leads to polypharmacological effects that in pathologies that can be so complicated, like neurodegenerative diseases or neuroinflammation. I mean, there are multiple components. So it's really hard to say which of them will be really the ones that at the end reaches the patients. In a way, all of them, they do have a chance. For example, in the case of the MAGL inhibitors, the ones that degrade the 2-AG, I mean, MAGL is the enzyme. In this case, for example, what they are also trying to do to avoid this tolerance is to use inhibitors that they are not always inhibiting the enzyme. It's just for a brief periods of time. And in this way, you can control how much 2-AG is really being staying for continuing activating the receptor, so trying to avoid tolerance. So this is still also in the clinical trials, so they are still trying to develop for clinical trials. Roche has a big project on that. So at the end, it's just a matter of seeing which of the different strategies will be efficacious in humans, because from this, of course, it's always hard to fully correlate what we see in animal models, in cells, to what is really happening in humans. From the theory and the information we have, all of them, they do have a chance, and they might have a chance in different pathologies. Might be that, for example, in the case of inhibitors of anandamide degradation, is probably mostly associated to anxiety disorders. In case of 2-AG, is in general, the belief is that probably they will be good for neurodegenerative disorders. In the case of the inhibitors of the transport, I mean, our current knowledge tells us that they could be good in both spectrums of diseases. But yeah, I mean, it's a long way to get to test them and to see really which ones are efficacious, because at the end, that's the main point, right? Which one will be efficacious. But according to the theory and all the knowledge we have so far, all of them, they do have a chance, and potentially all of them, they will be better than using the direct agonist to the receptors.
Okay, thank you. So you mentioned that there are some clinical trial currently going on with this kind of compounds. But regarding your specific work in your lab, you are testing this compound like in preclinical models, in a cellular model, or what kind of study are you currently going on?
Yeah, for the inhibitors of the transport, we have evaluated different mouse models associated to anxiety, models of anxiety, models of inflammation. We also did one of the latest publications we have is with a model of multiple sclerosis. I mean, there is neurodegeneration there and inflammation, and we have seen that they are quite efficacious. We also have assessed pain in general, a pain associated to inflammation. And in terms of in vitro models, we have tried to also look for a broad spectrum of inflammation or just general modulation of the system. And yeah, it seems to work. We have quite promising data with the molecule that is from the next generation that now is currently going to clinical trials. And yeah, the first approaches will be associated to neuropsychiatric disorders. And how it's called, these diseases that they do not have a quite attention. I forgot the name of these diseases. It's a medical need to get new pharmacological strategies to target these diseases.
Orphan.
Yeah.
Is that the word we're looking
orphan.
Sorry.
No
Yeah,
worries.
that's
I enter to interject with a couple of questions actually they came out from the audience, and actually thank you. And I just remind you that time is running, so we have the last 10 minutes with her, and so if you want to get your questions replied, this is a good moment to put them in. One, I'm gonna try. Yes, one is coming from Eva, and she's asking for those results from the clinical trials we just mentioned. By the way, I wanted to correct myself. I said Portugal, but it's the company that was Portuguese. The actual trial was run in France. And yes, they can be found on PubMed, and I will make sure to follow with a link as soon as we finish this live. But this question is actually really for you, and I'm delighted that Adrian is with us today. He's one of the smartest people I know. Thank you, Adrian, to be with us today. And there it is, a very smart question, actually. So he asks, by endocannabinoid transport, do we mean transport through the membrane, or are we talking about cannabinoids bound to something like a fatty acid binding protein when moving in circulation or inside the cytosol?
Yeah, in this case, we are talking about transport through the membrane. Yes, indeed. Yeah, I didn't mention about these other, it's better called carriers. I mean, there are certain proteins that are in the extracellular compartment or the intracellular compartment, then, yeah, of course, can carry the endocannabinoids through these very aqueous medium, because of course they don't have, or they don't like so much water. And yeah, in case of the intracellular carriers, there is the fatty acid binding proteins. Yes, a lot of research has also been done on that. But no, we are talking about a very different protein, which is at the plasma membrane. And yeah, exactly, it just is in charge of the transport across the membrane. So then the endocannabinoids can later on be carried by the fatty acid binding proteins or other proteins, because it's not the only one. There is quite a few that can also be intracellularly transported endocannabinoids to intracellular targets and so on. I hope I answered the question enough.
Thank you. In case Adrian drop us another question, otherwise I'll leave the floor back again to Fabio, and I'll just repeat by the end.
Yeah, regarding this kind of transporter or carrier inhibitors, did you test if they also are active on other transporter or enzyme or receptor, or do you think they are specific for these transporters?
Yeah, I mean, indeed, yeah, I suppose you mean the inhibitors of the transport that we are evaluating. Yes, yeah, we actually did a very quite extensive screening. We also test, for example, the fatty acid binding protein, the number 5 particularly, and there was no significant binding or effects associated to these molecules. We also tested like about 45 proteins, transports, and receptors in the CNS. There was no significant binding or activity modulated for all of this. Of course, we also test them into the enzymes that degrade anandamide or 2-EG, also not just the main ones, but other ones. And yeah, so far, I mean, it really seems to be a very selective molecule at a very high, very low concentration. So it has a high affinity for our target. And yeah, we really also try to take care of potential off-target effects that can also affect the development of this molecule. And so far, it's quite clean, thankfully.
Yeah. So, and probably some effect can only be seen like in preclinical or clinical study. But
sure.
I think it's very promising, your research, because and this demonstrates also that the research in the field of the cannabinoids and the endocannabinoid system is always developing and ongoing. And related to this, apart the work on this kind of inhibitors, there are other projects that you are currently working on in your lab that you would like to share with us?
Yeah, sure. I mean, we normally also have collaborations with other groups. We are also constantly, well, not constantly, but regularly screening for other different ligands to the receptors. Yeah, in terms of safety, also just for research, understanding all the different structures that can target or not the cannabinoid receptors. There is also a publication we have some years ago about a natural product present in moss, the cis-perotatinene. We were also trying to actually understand some additional effects we saw with this molecule, because this is also a molecule similar to THC that is also an agonist to both receptors. But we observed some differences, some pharmacological differences in the model of multiple sclerosis. And yeah, we are also trying to understand which other targets are also responsible for these effects, and how can we understand better these kind of molecules. We are also recently involved in a study associated with sleep, trying to differentiate actually THC versus CBN, cannabinol. And because there is also the belief that CBN might also be useful for sleeping disorders, but there is actually not real or substantial research. There is just a few very old papers that are actually not really evaluating sleep. So we are also trying to understand this also for potential use in humans.
Are you testing this in preclinical models?
First in a preclinical model. Actually, the data we have so far is quite promising. We are trying to gather some last information to also publish it soon. And yeah, I mean, everything really points to a certain difference that can be also leading towards a more beneficial use of CBN for sleeping-related disorders compared to THC. And
please go on.
There is a few other things that we also do around, but so far nothing comes clearly to my mind.
Okay, you are involved in a lot of projects. I knew you from your publication that are very relevant in this field. So maybe my last question will be, among all these compounds that you talked about, that you are aware of in that modulate the cannabinoid system, which one do you think will have the best potential to be put in therapy in the near future, according to you?
I mean, if we consider the near future as for the ones we have more information, perhaps the inhibitors. But I mean, if we do consider all the research we have so far, we actually do hope that the clinical trials that will be performed with the inhibitors of the transport can already tell us some information, because it might really be a much better alternative compared to the inhibitors of the degradation. But of course, all of them will have a chance to see perhaps which one will be the more efficacious. But it's a matter of time to understand which one it really proves efficacy, which is the most important part, of course.
Okay, maybe we should invite you when this result will come into public, so we can discuss something. We hope a novel treatment for a kind of pathology. So maybe Viola would like to add something.
Sure, I'm fascinated. I think we definitely would love to have you here again, giving us some updates. I think we have a question from Dennis that is quite on point, and he's wondering whether he understood right that your lab has developed a specific drug for the endocannabinoid system, or designer cannabinoids, or you're just testing.
I mean, I'm not familiar with the concept of threats, actually, but I mean, I don't know if in general make means for structures associated to the inhibition of the transporter, perhaps. I don't know, because, I mean, if that is the case, I mean, we have evaluated really quite some different structures to see if they can inhibit the transport process. And I mean, the second generation is now has been taken by a spin-off company from the university, Cannabinoids Therapeutic, and they are the ones that are actually taking this molecule to the clinical trials. But I'm sorry, I'm not really familiar with these terms of threats.
And I believe that they're simply just it's a way, my understanding, to call just simply small molecules that act as chemical actuators, if I'm not wrong, right, Fabio?
If I remember,
otherwise, Dennis, give us some more. Like what are we supposed to be talking about?
It's a new technique to evaluate like neuronal pathways, behaviors, or which kind of receptor is associated with a certain partner. I'm not in the dread, so I don't know how they work, but they are used to evaluate neural pathways mainly. So I think this was the question by Dennis.
Yeah, I mean, we are for sure trying to understand how these transporter affects overall the whole system, and yeah, we are trying to use some tools, genetical tools, to try and to understand this, how the endocannabinoid levels are modulated when this transport is not there. Of course, we also have to try to understand with these small molecules, these inhibitors by itself, and from the first generation, the second generation. Sorry, I'm just checking that he wrote the meaning, designer, receptor exclusively, designer for designing drugs.
Yeah, thank you to give us the acronym so we all have it clear. Designer receptor exclusively designed, I would say, for designer drugs, right?
Yeah, I mean, at least us, we are not working particularly on that then. Yeah, I think there has been some works with the CB1 and CB2, CB1 for sure, CB1 receptor, also to see, especially for modulation of calcium levels and how the endocannabinoid levels change. But us personally, our group, no, is not really involved with this kind of research.
Thank you.
Okay,
and
thank you,
on the other
Ines.
side, thanks everyone that participated to this very interesting live. As you can tell, half an hour covers, just brushes the surface of the many things that they're doing. I believe we will perhaps have already scheduled another appointment with someone else from the same lab, because they're very active, as you might have imagined, from many years. So there is quite a lot to touch on, but we'll update you with that. And I will want to just to thank again Ines to be here and share some science with us today. Thanks again. Thank you everyone participating today. And of course, we invite you as usual next Tuesday, Central European Time, 5:30, and we'll continue to spread awareness and education on any topic endocannabinoid system and cannabis associated. So thanks again for being with us today.
Thank you. Thank you for the invitation.
Thank you, Ines. Thank you, everyone. And see you next week with another appointment with the Cannabiscienzias live on LinkedIn. See you next time. Goodbye.
Ciao.
Ciao.